spoken a lot about muscles, origins, insertions, attachments, movements. Even in the lab, we've identified a lot of muscles and their location across joints. And so this is something we know. A muscle acts across the joint, perhaps you look at the elbow joint. What muscle can we identify acts across the elbow joint? Come on, we did the upper limb muscles. What's one muscle that acts across the elbow joint? What the chief flexor of the upper limb of the arm? The biceps brachii. Good. So the biceps brachii is inserted in the forearm, but then its origin is in the arm. And so its action is across the elbow joint. And when it contracts, it's going to bring about flexion of the elbow joint. Right? Now, in order to coordinate muscle activity, in order to help keep the muscle movement synchronized, which means that only one group of muscles are acting at a time, we have certain receptors in the muscle to help coordinate that activity. We also know about origins and insertions, and as a result, muscles are pulling and not pushing. Okay, so a muscle is fixed at its point of origin, and then it's mobile at its point of insertion, which brings about those actions that we're talking about. So flexion, extension, and so on and so forth. So a muscle can only pull, but it cannot push. Now, muscles are antagonistic in their action. This is something we've already discussed as well. We have the flexor compartment, which antagonizes the extensor compartment, and so on and so forth. And the reason that muscles are antagonistic, again, is to help coordinate movement. If you're trying to flex the forearm, you need to activate the flexors, which also want to deactivate or inhibit the extensors. Okay, so muscles are antagonistic in that way. A good example which is shown here is the biceps, which are the flexors. They decrease the angle across the joint. Whereas on the posterior side, we have the triceps, which are extensors, and they will increase the angle across that joint. So we said flexion, decrease the angle, extension. Now today we're going to talk about two receptors that are found in muscle, the muscle spindle, as well as the Golgi tendon organ. And the purpose of these receptors, again, is to coordinate that smooth, seamless movement as muscle contracts. The muscle spindle is a receptor which detects the change in length of the muscle. Okay, so any changes in length are picked up by sensory nerve endings that are present within the muscle spindle. Whereas the Golgi tendon organ is not a part of the muscle, it's actually a part of the tendon, and this detects any tension that's put on the tendon. We'll talk about how this can be protective for the muscle. So we have the muscle spindle detecting change in length, and the Golgi tendon organ detecting changes in tension to the tendon. All right, so when we look at the muscle spindle, these are basically specialized parts of the muscle, and they are unique in that they can pick up changes in length because of these sensory nerve endings that they're associated with. Okay, so we have two components to the spindle essentially. Intrafusal fibers. These are those unique specialized fibers. They're the contractile parts of the spindle. Right? And they're going to be detecting any changes in stretch or length in that muscle. They're innervated by gamma motor neurons, and we'll see the afferent fibers that they're associated with. Whereas the extrafusal fibers, these are the typical muscle fibers of that muscle, muscle belly, that muscle. Okay, so they're not specialized in any way. They're not associated with the afferent fibers that the intrafusal part of the muscle is associated with, but they're just the regular, normal muscle fibers. Their job is to contract, but they're innervated by a different type of motor neuron, alpha motor neuron. Okay, so that's just your regular old muscle fibers. When you look more closely at the spindle, it's associated with the annulospiral ending and the flower spray ending. Now, a lot of students usually get tripped up by the fancy words here. These are simply afferent nerve endings.
Going from the sensory organ to the central nervous system.
Correct. So that's communication from the receptor or the sensory organ up to the central nervous system. That's your afferent fiber. So there are two types here. The 1A afferent fiber associated with annular spiral endings, and the type 2 afferent fiber associated with flower spring. And these names are just descriptive of how these nerve endings associate with the muscles. So let's see what that looks like before we continue. Any questions so far? So let's talk about the structure of the muscle spindle, and then we'll relate it to an example of reflexes. Okay, so as we mentioned earlier, within muscle, we have our typical extrafusal fibers. These are the contractile fibers of the muscle itself, whereas internally, we have this modified region of the muscle that's called the spindle, and this is essentially made of the intrafusal fibers. And the nerve endings that are associated with these fibers. Okay. Now, those nerve endings we mentioned already are the annulospiral. And as the name implies, this nerve ending interacts or wraps around the muscle spindle or the intrafusal fiber, sometimes called the central bag. This region is sometimes called the central bag. And then the flower spray ending, runs off to the side and usually attaches to the distal part of that intrafusal fiber. Okay, so this is your flower spray. Ending, and then your annulospiral ending. Okay, and together these three structures, so the intrafusal fiber along with the two sensory nerve endings together make up the muscle spindle. Okay. Alrighty. So let's talk about these fibers one more time. We said that this was a type 1A afferent fiber, whereas the flower spray was a type 2 afferent fiber. Alrighty. Now we also mentioned that the efferent fibers differed as well. So the efferent fibers to the extrafusal fiber were basically alpha motor neurons. And then the efferent fiber to the intrafusal fibers were gamma motor neurons. We'll talk about why this is important. This difference is important in a minute. All right, we know that alpha motor neurons and gamma motor neurons have a different speed of conduction. Okay. Alrighty, and so this is the structure of the muscle spindle. Let's talk about an example of how this muscle spindle offers protection. How does it coordinate muscle movement? Okay. So the whole purpose of the muscle spindle is to detect changes in the length of the muscle, and then it's going to bring about a reflex action of contracting or shortening that muscle. So basically the muscle stretches, and that is picked up as a change in length. That is going to send a signal to the central nervous system on afferent fibers, excuse me. And so let's put our signal here. The CNS will then send a reflex signal back out to the same muscle group that's going to bring about muscle contraction. Okay. So we have the spindle detecting the length at first. Okay. Now, the way that the muscle, the sensory part of the muscle, the muscle spindle, the way that it sends signals to the spinal cord, right, to the central nervous system, is by changing the frequency of action potential. We talked about this during the first part of the semester. If you remember, we talked about frequency coding. And so to increase or amplify a signal, we simply increase the frequency of action potentials that are being fired. So increasing the action potential frequency here is how this signal is transferred. And this is transferred by those afferent fibers that we talked about. Alrighty. Alternatively, we have efferent fibers that are bringing back that response from the spinal cord. So this is a reflex. It's not going to go to your higher centers. It's simply going to go to the spinal cord here. Alrighty. All right, so the afferent fibers that carry that initial signal to the spinal cord are going to be your two fibers we spoke about, the 1A afferent and the 2 afferent, whereas the fibers that are bringing back that response to make the muscle contract are your typical motor neurons we've been speaking about so far. So your alpha motor neuron and your gamma motor neuron. Now let's talk about an example of how this muscle spindle brings about protection of the muscle. So think about you being at the doctor's office, you're sitting on the examination table, and your doctor is doing all of your tendon reflexes. So your legs are dangling off the edge of the table, right, relax. And your doctor strikes your patella tendon with the reflex hammer. Now striking your tendon with the reflex hammer is going to bring about a dent in the tendon, which is basically the stretching. Okay, so that's going to trigger this reflex. That initial dent caused by the hammer stretches that muscle, and the spindle, which is inside the muscle, picks up that change in length. Okay. So what do you think is going to happen at this point? After the muscle detects a change in length, what happens based upon what we've spoken about? Is it going to increase action potential frequency or decrease action potential frequency? It's going to increase. It's going to increase the action potential frequency, which immediately sends a signal to the central nervous system, which sends a reflex signal back out to this very same muscle group to cause that muscle to contract. Right? This is a protective measure to protect overstretching or overexertion of that muscle. When it stretches, when there's any change in length, immediately the muscle wants to contract. Okay, so when your doctor taps your patella tendon and wrinkles or puts a dent into that tendon and stretches it, the next thing that happens is what? What's the next action that happens after you strike that tendon? The muscle contracts, right? The quadriceps muscle, they contract, and so they shorten. And that brings about the kicking action that you get as a result of that reflex, of that testing. Okay? So that reflex is a good example of the muscle spindle and how it protects the muscle. So changes in the muscle length, stretching of the muscle brings about immediate contraction as a protective mechanism.